Hello, my name is Soumya Raychaudhuri, and it's a pleasure to be here today to talk to you about some of the work we've been doing in using genetics and genomics to define the molecular mechanisms of rheumatoid arthritis. Just by way of introduction, I completed my bachelor's in mathematics and biophysics at the University at Buffalo in 1997. I went on to Stanford University, where I completed my medical and doctoral degrees in 2004. In 2006, I completed my residency in internal medicine at Brigham and Women's Hospital and at Harvard Medical School. In 2010, I completed my clinical training in rheumatology at Brigham and Women's Hospital at Harvard Medical School. And at the same time, I also completed my postdoctoral training with Mark Daly at the Broad Institute and opened my own lab at Harvard Medical School. 2018, I was appointed full professor, and in 2019, I was appointed institute member at the Broad Institute. I have had the pleasure of mentoring several talented Japanese rheumatologists, including Yukinori Okada, Chikashi Terao, and Kazuyoshi Ishigaki. The main focus of a lot of my work in our lab is in rheumatoid arthritis. This is a picture of Pierre-Auguste Renoir, the famous French painter who was affected with rheumatoid arthritis. As you can see here, from the blow up of his hand, at this stage in his career, his hands were so damaged with rheumatoid arthritis that he was no longer able to hold a brush. Paintings like this one on the right, his self-portrait, were painted with the brush strapped to his wrist. Rheumatoid arthritis has a genetic component to it. One way to understand this is to look at sibling risk. We simply ask the question, what is your risk of rheumatoid arthritis if you have a sibling, a first-degree relative with whom you share half your genome, who has RA, versus the population risk? And as you can see, for many autoimmune diseases, your risk is dramatically increased if you have a sibling affected with with the same autoimmune disease. And rheumatoid arthritis, you see the same thing. This suggests a strong genetic basis for rheumatoid arthritis. Work done with Yukinori Okada, with Robert Plenge and Kazuhiko Yamamoto, helped us identify in 2014 risk alleles for rheumatoid arthritis in both Japanese and European populations. We're now in the process of doing a much larger study to identify risk alleles in many populations throughout the world. However, risk across all loci is not equal. There's one locus here, which you can see, which has the greatest amount of risk. Here, each dot represents the significance of a SNP association in different points in the genome as plotted on the x-axis. And what you can see right away is that the MHC locus confers a dramatic amount of risk compared to all of the other alleles in the genome. So in this talk, I'm going to explore the genetics of rheumatoid arthritis and talk about how we've been using functional genomics in different ways to understand how these different alleles are conferring risk of RA. First, I'm going to talk about defining HLA alleles and their effects. Second, I'm going to talk about annotating the genome for key regulatory elements. And third, I'm going to talk about defining causal SNP alleles and their functions. I'll start by talking about HLA alleles and their effects. So in 2012, we did a fine mapping study and identified the key positions in HLA-DRB1 that drive rheumatoid arthritis risk. And what we found is that position 13, 71, and 74 in aggregate, aligning the P4 pocket, explain a dramatic amount of the risk within the MHC locus. And in particular, position 13 explained about 90% of the HLA-DRB1 signal for RA. So position 13 is very important for rheumatoid arthritis risk. Subsequent studies show that the same position is really important, not just in European populations as shown here, but in Asian populations and African American populations. So, so that brought about an important question. How does the HLA interact with an antigen, perhaps citrullinated peptides, to trigger autoimmunity? So what we know is that there are antigen-presenting cells which express the HLA. When an antigen is presented, it might be recognized by a very specific T cell receptor, by the TCR, expressed on a T cell, and that results in activation. So antigen specificity really comes from two sides. So first, there's the HLA. So commonly we think that HLA risk alleles confer risk because they bind that antigen, but also by the T cell receptor. And specifically in the T cell receptor, it's the CDR3 region of the T cell receptor that expresses the antigen-specific region of the TCR. So how does RA confer risk? Well, there's two possibilities. One is the central hypothesis, which is on the left, and the other is the peripheral hypothesis on the right. Now, when we think about RA, it's usually the peripheral hypothesis that we talk about, that there's an antigen-presenting cell. It expresses an HLA, a high-risk HLA, and that high-risk HLA wants to bind that pathogenic antigen, perhaps a citrullinated peptide. But there's another possibility, which can also happen, and both can be true, which is the central hypothesis. In the central hypothesis on the left, what you see might be happening is that you have thymic epithelial cells expressing the HLA, and that's binding thymic peptides. Now, that's then presented to T cell receptors. Now in the thymus, if there's no binding, those T cell receptors don't survive. They don't make it to the periphery, and they don't become part of the T cell repertoire. On the other hand, if they bind too strongly, they're selected against and they're removed because they're at risk of causing autoimmunity. So only a certain subset of the randomly generated TCRs then make it out into the periphery. So under the central hypothesis, the idea is that the HLA is actually influencing the shape of the T cell repertoire by working through selection. There's already a lot of evidence for the peripheral hypothesis. In particular, I'll point out the work of Ranjani Thomas and Rosjan in this JX Med paper and a subsequent paper, where they really showed that the HLA structures are able to effectively bind citrullinated peptides, and that the HLA risk alleles bind the citrullinated peptides better. But is there evidence for a central hypothesis? So TCR composition, especially in the hypervariable CDR3 region, is critical for antigen binding. TCR composition is modulated in the thymus, as we talked about. Does HLA composition influence TCR composition? V gene selection in humans, we already know is influenced by the HLA, but the alleles driving autoimmune risk are different from those alleles that have been previously published. So we asked the question, does HLA genotype influence the CDR3 composition, the CDR3 region of the TCR, that region that's so important for antigen identification? If so, it might be through variable thymic peptide selection. So the strategy here is TCR sequencing. So new technologies have now made it possible to apply targeted sequencing to define TCR across a wide range of cell types to infer TCR sequences from RNA-seq data. And the kind of data that you get is that you get for many individuals, for any individual that you sequence, you get many TCR sequences. And so the strategy is that to test the relationship between the HLA and TCR. If genetic mechanism of HLA and autoimmunity has a central hypothesis component, then HLA should be associated with TCR features, particularly in the CDR3 region. And again, just to emphasize, antigen-presenting cells on the left here express MHC, which binds antigen. And the CDR3 region is the region we're going to focus on in this talk, because that's what gives the specificity to antigens. There are V regions as well. There's an alpha and a beta chain, but the CDR3 region is where the maximum amount of specificity emerges from. We worked with two public data sets, a discovery data set and a replication data set, as illustrated here. And the two data sets were different. On the left, the discovery data set had data from 666 individuals of unknown ancestry. This was a targeted sequencing study, so there was data on on the beta chain in almost 200,000 TCRs per individual, and the HLA was genotype. In the replication data set, we had 169 individuals of European ancestry. We had RNA-seq data. We had both access to alpha and beta chains, but in this context, this was only naive CD4 T cell data, and we had a smaller number of TCRs, only 1,800. And all of the work I'm presenting here was led by Kazuyoshi Ishigaki. And Kaz developed a specific analytical strategy, as illustrated here. So he had looked at every individual and looked at every TCR CDR3 position, and for each of the 20 amino acids, he said how many of those TCRs have this specific amino acid. So for example, for individual A, you can see that 3 out of 9 TCRs have an alanine or an A. In individual B, you can see that the number is higher, it's 6 out of 9. And so in that way, we developed for every amino acid a particular number for every individual, which was the fraction of times that amino acid showed up. First question we asked is are all these measurements independent of each other? And if you look at that CDR3 region, on the left, you see the V gene cluster, and on the right, you see the J gene cluster. So some of these CDR3 positions on the left are influenced by the V gene or encoded by the V gene, and on the right are encoded by the J gene. But in these middle positions, you can see that they're really random. And as you'll recall, when the TCR beta CDR3 is made, there's a region which is random, and that's that random region that gives a TCR its specificity. And in fact, we see very little correlation in between these positions. And then to identify whether there was association, Kaz used the statistical strategy here, where he asked, are the 20 amino acid usage ratios at a specific CDR3 position influenced by HLA DRB1 alleles at a specific position, for example, position 13 or position 11. And then he defined a linear model, and in R, using the MVLM package, he was able to assess significance. As you can see, we tested roughly 10 CDR3 positions and 309 HLA positions. So we had over 3,000 tests. So we had to do multiple hypothesis testing correction to identify significance. When we looked at the data, we identified a highly significant site at position 13, as you can see here, with a p-value of less than 10 to the minus 137. Here's another view of it. So across the top, I'm looking at the different HLA DRB1 positions, and across the left, we're looking at the different CDR3 amino acid positions. And what you see is that the greatest variance explained is really in position 11 and position 13, showing that these positions have the strongest effects. And you can see position 109, 111, and 112 are most strongly associated between CDR3 and HLA DRB1 position 13. Now, this is very interesting because, as I mentioned, position 13 is an important position for RA. It's also an important position for other diseases. For example, in type 1 diabetes, we'd previously shown that position 13 is an important driver of type 1 diabetes risk in HLA-DRB1. So where are these CDR3 QTLs coming from? We considered several possibilities. One possibility is that that it's hardwired in the genome, that is there's something about how the genome is encoding HLA and TCR, that they're co-regulated. The other possibility is that there's antigen experience in the periphery, that certain TCRs, because they're running into viruses or running into antigens as individuals age and develop memory T cells. It's that experience out in the periphery that's causing this sort of association. Third is thymic selection. So first, let's look at hardwiring in the genome. To do this, we considered immature T cells, and our strategy was to look at non-productive CDR3s. So non-productive CDR3s are these CDR3s that are produced where they have a stop codon or a frame shift problem. So these TCRs are never expressed in protein, but do exist in the genome. But they're not under selection because they were never expressed. So if HLA CDR3 associations are hardwired in the genome, then non-productive CDR3 should also have HLA associations too. In our analysis, we looked at HLA CDR3 QTLs in non-productive CDR3s. And in fact, look at these dramatic results. For the productive CDR3s, you see a dramatic association. For the non-productive ones, you see no association whatsoever. Now, we recognize that non-productive CDR3s were only 18% of the TCRs. So we downsampled the productive ones. And again, you see there's a dramatic difference in the signal, suggesting that, that the influence of HLA on CDR3 composition was happening after the DNA level. It was happening at the protein level after the TCRs were expressed. Here's another way of looking at the same data. These are the z-scores, which show the individual HLA associations. And as you can see, for the productive scores, you get these very significant z-scores of -10 or +10. Non-productive, you see no association whatsoever. The z-scores range from +1 to -1, very typical of random noise. So we ruled out the possibility that HLA CDR3 connection was happening at the DNA level or in immature T cells. What about antigen experience in the periphery? So first, we considered clonal expansion. So as you know, when naive T cells are made to interact with an antigen and are stimulated, they proliferate and become memory T cells. So one possibility was that perhaps certain CDR3 features allowed for this to happen better. So antigen experience converts naive T cells into memory T cells. Memory T cells expand clonally. Our analysis treated each clone as a single event. But if the associations are driven by HLA antigen presentation in the periphery, then accounting for clone frequency should augment associations. So the analysis here was to change the analysis so we upweight clonally expanded CDR3s. Now here's the interesting result. What you see in the pink is that the association for non-clonal expansion is much stronger than the one for clonal expansion, suggesting again that clonal expansion is weakening the association, not strengthening it. Here's the same data. Again, you can see that the analysis without clonal expansion, that is treating each clone as an individual unique event, is much more dramatic than the one with clonal expansion, suggesting that this is probably happening in naive T cells before proliferation. Another very simple way to consider whether this is coming from memory cells or naive cells is looking at the naive T cells themselves. And as you'll remember, we had a replication data set on the right here, which was all from naive CD4 T cells. And in fact, when we tested our naive T cells for CDR3 QTLs, for HLA CDR3 associations, we observed very consistent effects as in our discovery data set, suggesting that these HLA associations exist in naive T cells prior to conversion to memory. So our conclusion was that thymic selection is the likely driver. How is position 13 mediating thymic selection? Here's a structural perspective. So what you see here is if you look at the positions where there's the strongest association, the distance between HLA and the antigen is very close at position 13, and the distance between CDR3 and antigen is very close in these strongly associated positions. So while position 13 and CDR3 don't directly interact, they have an interaction mediated through the antigen. So here's the three-dimensional view. And again, what you see here is at the top is the CDR3, at the bottom is the HLA site. Here's the antigen in the middle. So these are the distances between 109 and the antigen 13 that are actually really short. So the amino acid here ends up influencing selection of the amino acid in position 109. Here's that same data. Here what we did was we took 5 structures and calculated the pairwise distance between residues from HLA, the antigen, and TCR3, CDR3. We embedded these 5 structures in a 2-dimensional plot. And what we see right away is that position 13 and 71 are, again, close to the antigen, and position 109 and these other CDR3 positions are also close to position 109. So the hypothesis that we made is that indirect interaction between HLA sites and TCR are mediated through a bound thymic antigen. Do risk alleles for autoimmune disease favor certain TCR sequences? So here what we did was we went back to our rheumatoid arthritis data and remember that specific HLA-DRB1 alleles have specific odds ratios that we can take from the literature, and we can come up with an individual score, which is our HLA risk score. And then we can use that to go back to our TCR data and say what kind of TCR would be preferred for different diseases. So for rheumatoid arthritis risk alleles, you can see that that acidic amino acids like aspartate D and glutamate E are favored in that TCR. There are different patterns seen for type 1 diabetes and celiac disease. And in fact, you can look at properties, and what you can see is that for rheumatoid arthritis, negatively charged TCRs are important, whereas for celiac disease, not so much. But on the other hand, hydrophobicity becomes really important for celiac disease TCRs. So in conclusion of this section, HLA alleles influence autoimmune disease risk and also influence TCR composition. Specific HLA-DRB1 alleles are associated with specific amino acid substitutions within the CDR3 region. Key driving position is position 13, same position that is essential for RA and type 1 diabetes risk. It influences up to 10% of the frequency of amino acid residues within key CDR3 positions. TCR amino acid changes track with RA risk effects. Our data gives support for a role for the central hypothesis or thymic selection model component to RA risk, and the changes appear to lead to enrichment for very specific TCR sequences. These TCR sequences appear to be enriched for those binding autoantigens, but more studies are needed to confirm. So in this next part of the talk, I'm going to talk about annotating the genome for key regulatory elements. So now we're going to step outside of the HLA and look at the rest of the SNPs in the genome. So for these other alleles, and there's about 100 alleles associated in RA, we really don't know what the causal allele is. We have regions where we know they're SNPs associated, but we don't know the causal allele. So in 2013, we started to think about how we can use epigenetic data and functional annotation to focus our search for causal cell types. One approach that we used was epigenetic marks, and that's a powerful way to mark regions of the genome for gene regulatory potential and to identify plausible causal SNPs. We can assess if they are in potential regulatory regions, and the caveat, of course, is that regulatory regions are highly cell type specific. Our model is that you have a SNP, and that SNP is sitting in a regulatory region that's influencing a gene's expression. We can identify the regulatory region using genome annotations, such as histone marks or other epigenetic annotations. The key thing that we think is going on is that there's a regulatory protein that's maybe not able to bind as efficiently or more efficiently, depending on which SNP allele you have. So we started looking at enrichment of histone marks for all kinds of different cell types as listed here. And as early as 2013, we knew that CD4 T cell subsets were highly enriched for specificity for RA risk alleles, suggesting that RA risk alleles were acting through T cell enhancers. But the key to fine mapping is being able to define regulatory variation. And central so it's central for us to understand gene regulation in the cell type and right context for RA. How can we better define what causal regulatory regions might look like? Well, if we understood causal regulatory regions for rheumatoid arthritis, we would be a step closer to defining RA pathogenesis. So can we define precise regulatory annotations? That immediately became an important concern. So the strategy that we considered was, can we look at not just the genome-wide significant confirmed association, but can we take advantage of weak associations throughout the genome to think about polygenic risk? We know that there's thousands of alleles associated with RA. We know that we only know about 100 of them. So can we look at these thousands of associations, these weak associations, and ask where they are in the genome, because they may contain SNPs with weak evidence of association throughout the genome. These weak associations can be used to assess heritability of different individual genetic annotations. So if we could find a cell type specific annotation that explained a substantial amount of the heritability, we would be on the right track. So we wanted to find annotations in the genome that contain many of these weak associations, and perhaps those types of regulatory elements would be the ones that we would want to pay the most attention to as we fine mapped different risk loci. And obviously, if we knew those enhancers and regulatory elements, they would help us understand key features of pathogenic genomic regions for RA and also get to RA pathogenesis. So the approach that we used here was led by Tiffany Amaruta, and also in close collaboration with Kazuyushi Ishigaki. And this work came out in a series of papers in Nature Genetics and American Journal of Human Genetics. I'm going to talk for a second about ChIP-seq, which is a really important technology here. So ChIP-seq allows us to identify the location of transcription factor binding sites in a cell type. And you can see over here, the way this works is you have a particular transcription factor that's bound the genome. You sonicate the DNA and break it up. And then you use an antibody that identifies a specific transcription factor. So the antibody is really important. So if you have a good antibody, your signal is often good. If your antibody is not specific for the transcription factor of interest, then your signal is less good. Then you pull down those regions where you've had the antibody bind, and you sequence, and you map your reads back to the genome. And here you go, you see a peak where there's a lot of reads. And that's where that's where that specific transcription factor was bound. Now we wanted to focus on ChIP-seq because we were interested in transcription factors. We felt that if we wanted to find cell type specific regulatory elements, transcription factor data is essential, because transcription factors define cell types and cell states. So they would give us a set of regulatory elements that are really important in terms of function. So if we could identify where transcription factors bind, if we could identify the places in the genome they bind, then we could identify the key regulatory elements for key cell states. ChIP-seq can be used for specific transcription factors to identify binding sites, but it has a high false positive and false negative rate. So our goal was to define biologically motivated annotation taking advantage of transcription factor data. And we defined a strategy called IMPACT, which stands for Inference and Modeling of Phenotype-Related Active Transcription. The key concepts are that we had ChIP-seq data to define where transcription factor was binding, as I showed you before. We had data on many transcription factors, and we looked for the motifs of those transcription factors, that is the sequence signatures of those transcription factors. And then we used a logistic regression model to build a large-scale epigenetic data profile using hundreds of annotations. And the goal was to annotate the genome, and we focused on master transcription factors. So to illustrate this was that we imagine that we have a situation like this. So here we have the genome, and we have some regulatory elements. There's some that we're not really that interested in because they're maybe generic promoters or housekeeping genes. There's some that we're very interested in, such as these ones, these red ones on the left. These are the regulatory elements that are very specific for individual cell types. The one on the left we're really interested in because it binds a master regulatory transcription factor, as you can see in the red, and that's what we're going to use to train our model. This one in the middle, it doesn't specifically bind that transcription factor, or we don't see it in ChIP-seq data at least, but it's very similar since it binds a similar set of transcription factors. So now what we do is we use those motifs, those sequence signatures, to see where the transcription factors bind. And then we have ChIP-seq data to identify where the binding is happening. Then we use all kinds of public histone mark data, genome annotation data from many, many different cell types, and we're able to build a profile. And as you can see, this enhancer and this enhancer, they have certain common features. And now we're able to go back and annotate the genome using impact. Notice we're not going to annotate this regulatory element on the right. We do annotate peak 1 and peak 2 on the left because they have a similar genomic profile. So an example model here is for FOXP3. So we had a model, we had an impact model for FOXP3. We used 515 cell type specific genomic assay features. Our features included DNase data, ATAC data, FAIR-seq data, which captured open chromatin, and many other annotations, histone modifications, etc. We also, of course, knew where the intergenic regions were, intronic regions were, promoter regions were, and etc. And so now we're able to annotate the genome. So using FOXP3, this is the CTLA4 gene, and a gene very important for T regulatory cells, and the FOXP3 transcription factor is an important transcription factor for T regulatory cells. And what you see right away is we're able to identify regions with regulatory activity around CTLA4. What are the key elements that we're using? Notice that we're using a lot of T cell annotations and a downweights nonspecific annotation. So for example, CD4 ATAC, Th17 DNase. So these are the annotations that our classifier, our impact is using. Here's another example. This is the TBET impact score. Now we're looking at interferon gamma, an important gene in Th1 T cells. TBET is an important transcription factor for Th1 T cells. And again, you see a lot of regulatory activity in and around this gene, both in the promoter region on the right side and also throughout the gene itself. We compared impact to a whole bunch of other models to see how accurately we can predict transcription factor binding. And in fact, you see that impact in red outperforms many other computational tools in predicting where a transcription factor is likely to bind. To see if our strategy was good, we looked at EQTLs, and EQTLs are known to be enriched in promoter regions. So if this is the case, we expected that Pol2 ChIP-seq impact model would be most enriched for EQTL signals because Pol2 is the transcription factor that marks promoter regions. It might be, in fact, we speculated, better able to identify EQTL causal regions than standard promoter annotations themselves. And in fact, this is what we see. So look at the left. The Pol2 impact annotation is highly enriched for EQTL signals. The other annotations here, including transcription start site, which is a standard promoter annotation, is enriched, but much less so than the impact signal. So now back to rheumatoid arthritis. So using the genome-wide association data on rheumatoid arthritis. Um, we used our impact annotation and examined what was going on. We looked at 5 million SNPs, and we looked at data from European and Japanese individuals, and we excluded the MHC. Now look, what we observed is that T cell specific regulatory annotations explain 80% of the heritability for European and Japanese RA. We saw a signal for both Tregs, Th2, Th17, and Th1. This told us that the signal we were seeing for each of these different T cell annotations were roughly equivalent. We couldn't distinguish one from the other. They weren't independent of each other. But T cell specific enhancers were really important for RA risk. In fact, when we compared our annotations to other annotations, what you see here is in the blue, you have the Treg effect, which is large, and then we tried to look at other annotations and condition it away. And the conditional annotation is in red. And what you see is that our annotation, our impact annotation, consistently has the largest effect, suggesting it explains more genetic signal than these other annotations. But can it help us find causal variation? That, of course, was the idea we started out with. Here's two loci, CTLA4 and TNFAIP3. Now look, we can annotate with the Treg impact annotation, which explained the greatest heritability for RA. And now what you see is that there's individual SNPs that turn up sitting in peaks and have high impact scores. Now again, the Treg annotation isn't so different from the other T cell annotations, but we're able to pick out SNPs that we think might be causal. So we looked at many loci and assessed whether any of them are highly enriched for Treg impact annotations. We looked at high probability variance in high Treg impact regions. And what we observed is that for a number of loci, BOC2, ANC, RD55, CD28, CTLA4, TNFAIP3, IRF5, we're able to identify individual SNPs that are highly enriched for impact scores. So to summarize this section, impact is a strategy to combine many annotations to get genomic annotations that are specific for precise regulatory classes. Impact T cell regulatory annotations explain greater than 80% of the heritability of RA. We see consistent results in Japanese and European RA GWAS. They can help us fine map, as you'll see in the next section, causal variation for RA. But it's difficult to define which specific T cell state impact score is really the most useful in interpreting RA risk alleles. So now I'm going to start to talk about defining key causal SNP alleles and their functions. And this is work where we use data from rheumatoid arthritis and type 1 diabetes, led by Harmian Westra and Marta Martinez-Bonet, who did the functional work in Peter Nigrovic's lab. The work was a collaboration between geneticists who think about RA, such as myself and Peter Gregerson, but also from the Type 1 Diabetes Genetics Consortium with Steve Rich and John Todd. And all of these analyses were done in European data. And I just want to acknowledge my colleagues in the RA genetics field who contributed data from the RA immunochip study to this effort. The association framework, again, was simply looking at disease status and predicting it based on genotype. And of course, we included standard covariates that we include in genetic analyses for both RA and type 1 diabetes. Now, the important thing for this study that we did was we converted our SNP p-values to a Bayesian framework. So that allows us to convert the statistics about a SNP associations within a locus into probabilities that they're causal. There's some important assumptions that we make that I'll just go over. The first is that there's a single major effect. That's not always true. Sometimes there's multiple SNPs in a locus causing risk. We also assume that all variants in the genome have been captured. That's not always true because sometimes there are SNPs missing from our study. Each SNP within a locus is assigned a probability in this framework. And the most interesting loci are the ones where a small number of SNPs carry most of the probabilities. that is a single variant with a 90% of the probability for being causal is very exciting. Almost certainly the causal variant. But a locus where there's 20 variants, each with a low probability of being causal, is problematic. There's no clear winner, and it's hard to know where to focus functional work on. So when we analyzed rheumatoid arthritis and type 1 diabetes together, what we saw is that there was a small number of loci, one where there was a single causal variant, or two when you combined all the data, but several loci where there were less than 5 variants being causal, 7 in the case of rheumatoid arthritis. Here's the data displayed in another way. So at the outer ring, we have the RA alleles, type 1 diabetes, and combined. And I've colored the non-coding and coding alleles differently. And I've picked out the loci where there's a single SNP with greater than a 20% chance of being causal. So you can see, for example, the coding alleles pointing to PTPN22 or TIC2, where we know that there are coding alleles driving RA susceptibility. So coding variants are highly enriched for causality, about 17-fold enriched in RA and type 1 diabetes over background. We identified PTPN22, IFIH1, TIC2, SH2B3, and SIRPG as loci where coding variants have the highest posterior probabilities. And these were the same as what we knew about from previous studies. Now, this is an interesting one. DNase 1L3, it was known to be associated from previous studies, no association to type 1 diabetes. And you can see that there's a single SNP, R206C, that has the most significant statistic. Now, we convert that to a Bayesian probability. You can see that it has a very high probability of being causal, especially, and the other alleles have almost no probability of being causal. There's no independent association in that locus, and it encodes arginine to cysteine change in DNase 1L3. And this loss of function mutation is what's important for RA susceptibility. Loss of function allele puts you at risk for RA. It's also been shown that loss of function of DNase 1L3 is causal for systemic lupus erythematosus. Previous studies done by Yasuda and colleagues have shown that this same mutation causes enzymatic loss of activity for DNase 1L3. Where is DNase 1L3? It's not normally a T cell gene. In fact, it's a fibroblast gene. And what you can see is that DNase 1L3 is highly expressed in the lining fibroblast cells, as illustrated here. And in fact, if you look at some single cell data that we have, what you can see is DNase 1L3 is expressed in a subset, is expressed only in fibroblasts and only in the CD55 positive fibroblasts, which are the lining fibroblast populations. But you don't see any expression, for example, in NK cells or T cells or typical immune cells, suggesting an important role of this gene in stromal populations. Non-coding alleles. I'll focus for a second on CD28-CTLA4, which is associated with both type 1 and rheumatoid arthritis. CD28 and CTLA4 both important genes in T cell regulation. Here we found a single SNP with high probability. Now it's interesting because at first look, it looked like there was different SNPs for type 1 and RA. On the left, you can see for type 1, there's RS3087243, and on the right, there's RS1177011653. Now, when we analyze these SNPs together, what we found is that the two SNPs together are more strongly associated than any other pair of any other two SNPs in the locus. And this is what we think is going on. We think that the two SNPs are working together in both diseases, that in one haplotype, if you have the two protective alleles, you're at no risk. In the other case, if you have the protective allele, the A allele for RS3087243, you have an intermediate risk. And if you have the protective allele for both diseases, then you're at really low risk of RA and type 1 diabetes. And as I mentioned, we have a particular model where we think SNPs like this one are affecting how regulatory proteins are binding, and that they're sitting in regulatory elements as shown in the previous section. Altered binding may cause differential enhancer activity and then altered expression. So we assayed the function using EMSA and luciferase assays. Here's the data using a luciferase assay. Look at that. You see specific binding for this in this particular SNP, but only with the C allele and not the A allele in Jurkat T cells. Marta Martinez-Bonet conducted a luciferase assay and showed that, in fact, both alleles had increased luciferase activity, suggesting that this SNP is sitting in an enhancer region or a region with enhancer activity, but the C allele has more function than the A allele. Using promoter capture high C, we're able to show that SNP, there's an interaction of that SNP, not with the nearby CD28 gene, which we talked about before, but actually with the CTLA4 promoter, suggesting that this SNP, which is closer to CD28, is actually probably acting on CTLA4. So to summarize, it's possible to find map common variant loci. There are some important challenges, which is that they have weaker effects, they have non-coding effects, there's linkage disequilibrium and missing variation. Our approach was to identify likely causal variants and confirm them functionally. We identified causal autoimmune variation in CD28, CTLA4, DNase1L3, which I talked about, but also MEG3 and TNF-AIP3, which I didn't mention. DNase1L3 encodes a functional coding change. CTLA4 encodes induces a non-coding change that appears to alter regulatory activity. Cellular function still needs to be defined for each of these alleles. So in conclusion, rheumatoid arthritis is a typical rheumatic disease with risk alleles inside and outside of the HLA. HLA-DRB1 position 13 and other alleles shape the composition of the T cell repertoire. We believe that this occurs during thymic selection. To define the effects of non-HLA alleles, we need to define the key cell type specific regulatory elements driving RA. We are able to define these key regulatory elements by modeling transcription factor binding. CD4 T cell regulatory elements explain 80% of the heritability for RA. The next step is defining causal alleles in rheumatoid arthritis risk loci and linking them to function in these key regulatory elements. We demonstrate key examples where we are able to identify the causal alleles for RA. Finally, before I close, I just want to thank our various funding agencies from the NIH, multiple institutes within the NIH, particularly NIAMS and NIID. And also, I want to acknowledge my lab, where, as you've seen, members have contributed directly or indirectly to the science. Thank you.